Good morning, everyone. Welcome all participants to the third webinar organized by ISA Inc. in collaboration with Windrock International, the implementer of Be Safe or Building Safe Agricultural Food Enterprises project. Today's webinar is on animal biotechnology opportunities and regulations in the Philippines. I am Dr. Dora Romero Aldemida, Executive Director of ISA Incorporated, based in the Philippines, your moderator for today's webinar. The approval and commercialization of Aqua Bounty GM salmon in the US and Canada, and recently, the gene-edited sea bream and puffer fish in Japan send a signal that biotech animals could be on our shores in the very immediate future. Thus, the Philippines, in the very recent years, while under the pandemic, developed the Animal Biotechnology Guideline that shall oversee animal biotech research and development, importation, and commercialization. And we are proud to say that, once again, the Philippines is one of the first countries in Asia to upgrade its biotechnology regulation to include animal biotechnology. So today, Be Safe, ISA Inc., and through our experts, we aim to raise awareness and understanding of animal biotechnology in the global and regional perspectives, as well as the opportunities and regulations of animal biotechnology in the Philippines. Can we now show the house rules? So this webinar is part of a series of webinars on an array of topics about modern agricultural biotechnology applications and products in the Philippine context. The webinar will run for 2 hours. Next slide, please. All microphones are on mute, and questions should be entered in the Q&A function only. The chat function will be used by the panelists and for important messages to the participants. All of the presentations and the Zoom recording will be posted at the ISA Inc. website and are freely downloadable. So maybe a day or two, and you will see these materials at our ISA website. We also invite you to stay till the end of the webinar and respond to a post-webinar survey, and respondents will be given certificates. So I hope I don't see these questions and messages in the chat box again. So to open and start the webinar, let me now introduce Mr. Mark Hansel. He is the current Agricultural Attaché for the Office of Agricultural Affairs in Manila, having arrived in only last April 2022. So this is his first engagement with us on agri-biotechnology. He joined the Foreign Ag Service in 2017, following a 25-year career in the private sector, most of which was focused on international marketing and business management. His first post was as Deputy Director of ATO Shanghai from 2019 to 2022. He's a native of rural southern Idaho. Wow. Mr. Mark earned a Bachelor of Arts degree from Harvard University, where his concentration was on East Asian studies. Friends and colleagues, let us welcome Mr. Mark Hansel to give the opening messages.
Thank you so much. I appreciate that kind introduction, Dr. Romero Aldemite, and I am really honored to be here. Mabuhay, everyone. I am so happy to be in the Philippines. I'm so happy to be working on agricultural projects here, and very honored to be participating in a seminar with some of the people I've had a chance to meet. Dr. Mingala, I've had a chance to have some conversations with him. Dr. Clarete is a good friend of our office. So we really appreciate a lot of the good work that's being done here and are happy that we have a chance to participate in it. The Foreign Agricultural Service is the overseas international arm of the United States Department of Agriculture. And Here in Manila, we have a staff with 2 U.S. officers. Many of you have met my supervisor, Morgan Haas. He's on his way back to the States now, working on the harvest on his family farm. He's a legitimate farmer when he's back in the States. And our office, we are working on trade issues between the United States and the Philippines regarding agriculture, and anything we can do to facilitate know-how back and forth, including a lot of things about biotechnology. And it's a really exciting space to be in right now. To feed the world, and our job in the Foreign Agricultural Service is truly about feeding the world. It's about making sure that agricultural products go where they need to go, that agricultural know-how goes where it needs to go. And to feed the world in 2022 and beyond, we're going to need every available tool. It's a blessing to live in an era when we have discovered and we have been able to create technologies like agricultural animal biotechnology. There are a lot of benefits that come from this. Animal biotechnology is beneficial to human health in indirect and direct ways. I have a very good friend who had heart surgery a number of years ago, and he now has, as part of his human heart, a valve or flap that came from a pig. And this is just one example of the ways that animal biotechnology has helped to advance human health. Animal biotechnology also improves animal health. For many decades, farmers have been improving their herd's health through enhanced animal husbandry and better practices, better technologies, artificial insemination, embryo transfer, in vitro fertilization, genetic mapping, cloning. All of these things are part of biotechnology that has helped farmers enhance breeding and resulted in healthier herds. And that's an important thing here in the Philippines, especially where there are a number of particular climate challenges that maybe other parts of the world, that maybe we don't face in the United States. Biotechnology is a critical element in helping us develop more nutritious food, improved animal health conditions and medicines and diagnostic testing. It results in safer food for consumers. So food quality can be improved by introducing desirable traits into the livestock and poultry. And finally, animal biotechnology is an important tool to improve conservation and environment in animals. It helps introduce more environmentally friendly animals. It helps reduce waste. It helps shorten the growth period for livestock and poultry and improve the quality of the product that we get from that. So there are so many things and so much promise that we've already realized from this, so much of it yet to come. And the Philippines is a place that I think has so much more benefit ahead of it in terms of what animal biotechnology can do. So we are excited to be part of this. We are very happy to see things like today's seminar happening that further this effort, that help farmers and producers here to have access to better ideas, better technologies. And our office stands ready to help with this. We have already played a role in facilitating the beginning of the Be Safe project and its ongoing efforts. We're honored to be part of that. We look forward to being part of ongoing efforts and newly developed efforts in the Philippines. So we wish tremendous success to today's seminar. We hope that if you have any questions to which our office can respond, or ways in which we can be helpful, that you'll contact us. And in the meantime, all the best to you today. And thank you again for the honor of greeting you as part of today's seminar.
Wow. Thank you so much, Mr. Hansel. It looks like we are going to have a very fruitful webinar today with your inspirational talk, the opportunities that the Philippines could have through biotechnology, animal biotechnology, and your support to the Philippines towards this end. So with that, we'd like to first acknowledge, thank you very much, Mr. Hansel. We would like to acknowledge the presence of Dr. Marvin Villanueva, Chief of DA Livestock Biotech Center. Thank you, sir, for being here. And now we can proceed with the first talk. And this will be on global status and opportunities of animal biotech for food and agriculture, to be given by Dr. Carl Ramage, PhD, GAICD, Managing Director of Rautaka Solutions, Australia. He specializes in strategic and operational directions in biosafety and the commercialization of biotech products, including the development of regulatory strategy and the drafting regulatory dossiers for submission and assessment by competent authorities. And he has been doing this for many, many years. He is the chair of the La Trobe University Institutional Biosafety Committee and a member of the Agricultural Industry Committee of La Trobe University. He's also a former board member of the Association of Biosafety Australia and New Zealand. He's a very good friend of ISA, having been our speaker for several times already, even across the seas. So let's welcome Dr. Carl Ramage.
Thanks, Ola, and thank you so much for the opportunity to speak to the participants. And I'd like to echo some of Mark's words about the importance of biotechnology and the importance of communicating in events like today, to be able to share our experiences and to engage with as wider members of the community as possible to talk about animal biotechnology and biotechnology more generally. So I'll see if I can get my presentation to work. Give me a thumbs up, Ola. Is that looking good?
Yes, sir.
Wonderful. Thank you. Thank you. So let me see. We'll go to the first slide. Okay, so Mark touched on it in his opening address. You know, animals actually have served and continue to serve as an incredibly important role in research and development across the world. We've been using animals for a very long time to study a range of things related to human disease, to understanding how we can increase animal productivity, their health and well-being. And more recently, we've also been using animal models to deal with a global pandemic in the development of vaccines. So animals have played such an important role already, and they continue to do so. But what we don't see necessarily is biotechnology, animal biotech products in the marketplace. In terms of creating these animals, there are really two key approaches. One is we're able to use cell biology to manipulate animal embryos, and out of that create a genetically modified animal that can be used to study, for example, a particular disease, cancer, or maybe some behavioral elements as well. The other approach uses cells. So we're able to, with new technologies, take cells from an animal and grow them in the laboratory, and then we transform or modify those cells and then convert them into something that can then go back into an animal's embryo and lead to a new animal. So these are the two key pathways for the manipulation of animals in biotechnology. And then these two processes have served us very well over the last 40 odd years in research and development. But I guess the great challenge for us is, how do we then translate the use of some of that technology to address some more fundamental questions? How do we provide genetic improvement of food animals to meet the demand that we have for animal protein? And how do we do that in a way where we don't compromise the well-being of animals, or, as was pointed out by Mark, compromising the environment? We know what we want. We want food equality. How do we get it? We use the technologies and the tools that we have to change our paradigms and to address the challenges that we face globally and more locally. But then how much does it cost? We know that the term biotechnology, and in particular biotechnology in animals, does create some social uncertainty. We know that there are political agendas at play when we're looking at the movement of products in terms of trade, but also popularity. You know, our politicians like to be popular. And we also know that technologies and advances in science are often clouded by intellectual property and patents. And then for developers, there are some key hurdles associated with navigating regulatory uncertainty and getting the right approvals in place to be able to take a product to market. Fundamentally, and I guess this is a key role that the USDA plays, is they're looking for universal access. We want to make sure that when these products have been developed or these tools and technologies are available, that everybody has access to use those to deal with the challenges that they may face. So it was mentioned in the opening that when it comes to biotech animals, there's really only one that really stands out as a genetically modified animal product that's more recently made it to market, and that's the AquaAdvantage salmon in North America. And this has been genetically modified using one of those two processes that I showed in my second slide, which leads to an animal that grows significantly faster to a counterpart. So this means that there is an ability to increase productivity in salmon with lower inputs. And as we know, salmon is incredibly good for us in terms of a very good source of as an oily fish for omega-3 oils, for example. But there is one other animal that is also commercially available that has been genetically modified, and some of you may have seen these around the world, and these are the glowfish. Not something we eat, but an ornamental, something that serves the purpose of providing us with a little bit of health and well-being when we have our fish tank with glowing fish. There have been, however, attempts to try to bring other genetically modified animals to market over the years. But as you can imagine, when we start to deal with the modification of animals, that this does cause some concern to the community. And there have been a lot of roadblocks for commercializing genetically modified or genetically engineered animals. However, more recently, in the last few years, a new opportunity has come to light that offers tremendous potential for the advancement of animal biotechnology. And this is a technology called genome editing. Now, genome editing has been around for quite some time, and it was essentially like the invention of the telephone. And over time, it has evolved into something that is really quite special. Essentially, what gene editing does, it allows us to be able to specifically target an area of the genome and make a cut. And that is, they cut the DNA in one place, and then depending on which pathway we decide to take, we can allow the cell to repair itself, or we may, and in doing so, it may create a change. It may create a mutation in that particular place, and that mutation may be something that's beneficial. And I'll talk a little bit about that later on. The other option is that rather than allowing the cell to repair itself, we actually provide a set of instructions, and we say, we would like you to repair this way. It's not that dissimilar to having a word processing program where we go in and we do some corrections of our spelling mistakes. We were able to correct a word or a letter, and that's essentially what gene editing allows us to be able to do. So it's a very powerful technology, and it has a whole heap of different names. So you might hear gene editing, genome editing, new breeding technologies, precision breeding, genome editing, new improvement techniques. There's so many different names, but it's different to genetic modification. It's not the same as I showed you in those first two slides. It may use part of the technologies there, but it's not the same. It's complex and often difficult to explain. And that's a challenge for scientists. The other challenge that we have is deciding what it is that we want to edit. Because if you think about looking at a manuscript, a story, how do we change that story? What do we decide? How do we decide on what we're going to change? I showed you a couple of examples of what's commercially available in animals, the salmon and the glowfish. Those were decisions. There's a demand from the consumer for cheaper omega-3 high fatty oil fish, and there's a market for people that like to have fish that look nice and glow in the dark. Similarly, we need to make some decisions about what is it in terms of animals that we want. We want increased productivity. We want disease resistance. We want increased muscle. We want them to have a better behavior, a better temperament. We want them to be able to deal with climate variability. Do we then know whether we have the tools or I've identified targets that will allow us to deliver that outcome? And that requires a lot of science, a lot of understanding. And I think we'll hear a little bit later on of some of the tremendous work that some of the scientists in the Philippines are doing at the Livestock Centre to identify where there might be opportunities to use these technologies for a desirable outcome for consumers and the environment. So what we do know is that we have a vast array of variability that exists within livestock, for example. So when we start talking about cattle, we know that we have beef types and we have milk types. We know we have some animals that are regionally adapted, some that are disease resistant, or have some sort of resilience to particular stresses. So utilizing our science, we're able to try and understand what those traits are, and we're able to use whatever tools we have to value add. And that was, again, another message that Mark talked about. It's about value adding. It's solving problems and issues that we may face. But we have a lot of this opportunity for genetic improvement. And when it comes to using biotechnology, the potentials are quite vast. So if we look just at beef and dairy, there are a range of things that we can do there. And I'll provide a couple of examples shortly. In swine, there, again, similarly, a range of productivity, animal welfare, and disease resistance opportunities that are available because we understand those mechanisms and we have the tools to be able to address them. And we talked about, we know that there are some aquaculture products in the market now. So let's start with disease resistance. So we know that pigs in particular, farmed animals, are susceptible to a range of diseases. And in particular, there is a particular disease that costs the pork industry in the US quite a significant amount of money. But we also know that there are targets using genome editing, for example, to be able to address that issue. Another issue for pigs is African swine fever, which I know in the Philippines that you've had some challenges with as well. There are studies going on in Scotland where they're looking at why it is that a closely related warthog is resistant to African swine fever. And by understanding why the warthog is resistant, being able to use the genetics and the genome editing technology to be able to convert a conventional high-producing pig into one that is resistant to African swine fever. Using the same sorts of logic, understanding why it is that other animals are resistant to some diseases, and then being able to convert those genes to the same type in a productive animal. What we're finding in Australia, and I think the Philippines would be the same, is that there is a huge demand on our agricultural sector to ensure the welfare of our animals. And in particular here in Melbourne, where I live, more recently, we've even seen that live exports, for example, have been heavily condemned. And we've seen on the backs of our taxis and the backs of our buses, advertisements trying to stop the live export of animals. We know the public's very concerned about that. One of the practices that is undertaken in the livestock sector, particularly dairy, is the removal of horns. And so this offers us an opportunity using the technologies that we have to provide an animal welfare solution. So out of the US, a company, Saligen, out of another company, Recombinetics, provided a demonstration of proof of concept of being able to gene edit cattle that would normally have horns to no longer have horns. And there's a range of commercial activities that are happening in that space. That same technology, we did some proof of concept work here in Australia with out of those animals. And we generated a number of dairy cows that would normally have horns that were born with no horns. So a proof of concept that you can use a genetic technology, an animal biotechnology to solve an animal welfare challenge for animals that would otherwise require their horns removed. And that's what they look like when they're a bit older. So as you can see, these animals never developed horns, and therefore they never needed to have them physically removed. The other big concern from a welfare perspective is heat stress. And part of this is being highlighted by the fact that in temperate regions, like where I live down here in the bottom of Australia, we have dairy cows that are very, very high producers of milk. They can produce up to 30 litres of milk per day per cow. But if we were to take one of those cows that like a nice temperate climate as we have here, and move that up into the tropical, subtropical regions, those animals become quite stressed. And what that means is that they lose their fertility, they have less production, they end up with some metabolic disorders, and they're very unhappy animals. So how could we solve that challenge, but be able to satisfy the demand for dairy products? Well, we know that 2/3 of the cattle in the world actually live in very, very warm climates. And we know that there are a large number of animals that survive and live quite happily when it's warm. However, they don't produce nearly the same amount of milk. So if we're able to study those animals, work out what it is that makes them tolerant to heat, and then convert one of our temperate climate high milking animals to be more tolerant to heat, we could then move them into those regions and solve a challenge there. And in fact, we see that this is actually being undertaken. So, for example, this is an example of some animals that don't like the heat, and you can see that even at 28 degrees Celsius, they like to stand in the shade because they find it very, very difficult. However, we know that there are these breeds like the Brahmins or some other varieties or breeds of animals, for example, that are heat tolerant. So we can identify the genes associated with that and try to convert one of these animals to be more heat tolerant. And in fact, there's a company in the US that has already got animals on the ground where they have gene edited an animal that doesn't like the heat to be thermotolerant. Another example is using gene editing to be able to create castration-free pigs. So this is a practice that's undertaken in the pig industry. Male animals create an odor, and therefore their meat is not as desirable. This boar taint odor and the aggression that's associated with males is dealt with typically by castrating those males. But again, as you can imagine, that's not exactly a very nice practice, and it has a significant cost. So can we use gene editing to solve this problem? And the answer to that is yes, and there is some pre-commercialization studies already being undertaken in that space. Ola mentioned at the beginning of the webinar that there are some gene-edited fish already on the market. On the left-hand side, there is a red sea bream, and on the bottom there, we can see a tiger puffer. These are gene-edited animals that are currently available in Japan, and they were gene-edited to increase their growth rates. So you can see there that the puffer fish there, the size difference based on a gene editing approach. Similarly, on the right-hand side, there is a gene edited tilapia that's also been exempted from regulation and is about to hit the market, or is already in the market in Argentina, and I think similarly throughout the rest of South America. So we're starting to see now, some opportunities where we could create livestock with improved animal welfare, disease resistance, for example, and increased productivity. But we're already now starting to see some fish entering the market that have similar objectives. So some key messages is that there are numerous genetic engineering and genome editing targets that have been identified, and a lot of work that's being undertaken to characterize animals. And I think we'll hear a little bit about that from Claro. There are a lot of biomedical models that utilize animals to study diseases and other problems. We have some ornamental fish and some high growth rate fish that are now on the market, but nothing with legs so much as yet. But there are significant opportunities to improve animal health and welfare, and particularly in livestock. And by understanding how we might be able to use tools to address some of those issues is the area of a lot of study at the moment. I guess one of the challenges is that the commercialization of those products will require a clear pathway to market and a value capture for the providers. So there needs to be an element of product pull to the market and a solid market for a product that utilizes these technologies. Key question for a lot of people when it comes to biotech, not just in plants, but particularly when it comes to animals, is the question of whether they're safe. And in particular, when we talk about biotech products, we're talking about biosafety. Are they safe to the environment, and are they safe to us as people? And I think we can all imagine that there are certain products out there, or sort of animals out there, that are harmful to us and harmful to the environment. So the question is, if we edit or modify an animal, does that increase its potential to harm our environment or us as people? And our regulatory systems take a case-by-case approach to this to assess them individually on their merits. And in fact, and the Philippines has been a world leader in this, most countries around the world have established biosafety regulations and guidelines to be able to assess whether products of biotechnology are safe and safe for us to consume and safe for the environment. And this is all about risk management. But there are a lot of international guidelines and procedures that can be used to evaluate products for safety. Particularly for biotech animals, we're looking at identifying where there might be some hazards. We look at the characterization of those hazards. We look at exposure, i.e. how much of it might be eaten. And we look at things like toxicology, allergenicity, and characterize those hazards to a point where we understand the safety of the food product. And these guidelines have been around for a very long time, more than 20 years. So this is a well understood process, and I think the Philippines have been well placed to lead that across the Asia Pacific. And so when it comes to the addressing new technologies like genome editing, the Philippines remains at the forefront of development of international regulations. So some key messages here. Many countries have developed biosafety guidelines to assess the potential harm or risks associated with biotech products, and I'll use that term more broadly. Often the trigger for regulation is how we define whether something's been modified or not, to the point that it may provide something that is harmful. So in the beginning, I talked about genetic modification in the Atlantic salmon, for example, aquabani salmon, versus a gene editing for thermotolerance. What is the difference here? What is the trigger for regulation? How do we assess the safety of those products? What are the potential risks for people or the environment from genome editing or from genetic modification? When do we undertake an assessment or when do we require an assessment? So we also know, and we're not presenting it here, but there are many countries, and the Philippines are no different, using decision trees to be able to decide what requires regulation, i.e. where the risk is higher or not. And I think this has been a great opportunity to advance animal biotechnology towards commercial products compared to what we've had over the last 25 years. My concluding thoughts are these: that the role of biotechnology in agriculture continues to grow. We've been using animals for a very, very long time to study a range of problems, such in the biomedical sector, as well as in agriculture, but we've never really progressed animal biotech products to the point of commercialization. But with the new technologies, we offer some great opportunities to solve some challenging problems, but that in itself brings new challenges. Challenges for consumers to think about whether they would like them, challenges for governments and regulators on how they might deal with them, and challenges for technology providers to ensure they have something that's actually worth money. The environmental impacts and product pull are really driving the development of products that are consumer focused, i.e. that is that consumers do want them because they see that there is a benefit for the environment, there's a benefit for them. And in particular, when it comes to animals, that there's a benefit for the animal as well. Key element to this is regulation. And often we look to our regulators to be able to provide us with confidence that the products, the foods that we're eating are safe for us to eat and that they do not have a detrimental impact on our environment. Often it's the definition of whether something's modified or not that provides the foundations for whether regulators need to intervene. And when it comes to new breeding technologies, as I said before, this is a challenge for regulators, but we're seeing internationally a lot of pragmatic approach to being able to understand whether these products need to be regulated or not, and how they're regulated. I'll add one other point, and that is around stewardship of products. This is incredibly important and something that has been pivotal to the introduction of genetically modified plants, crops. Maintaining the product identity through the supply chain is critical to ensure that we don't have negative impacts on our imports and our export markets post entry to the market. So there's still a balance between managing risk and innovation, and this will be the ongoing challenge for the introduction of agricultural biotechnology products. But with that, I will finish up and thank the organisers and thank you for your attention, and look forward to some robust Q&A a little bit later. Thanks, Ola.
Thank you so much, Carl. And our audience really appreciate the many opportunities that you have provided, not only for the global perspective, but also for the Philippines. And you're mentioning that the Philippines is one of the forerunners on regulation. And we're happy that we already have about 74 participants. And now I would like to know, let's give a round of applause first to Carl Ramage, a virtual applause. Thank you, Carl. Now we can proceed with a few questions. This is through our poll. So we will have some poll questions, and we'd like to know how much you understand the presentation. So please, Pauline, answer or respond to the poll questions. The first one is, do we need animal biotechnology? Yes or no? Do you think that gene editing could offer useful animal products? Do you support the use of biotechnology in animal production? And would you eat animal products derived from biotechnology? You have a few, maybe 30 seconds to respond, and we can, okay, host and panelists cannot vote. So please, participants, you can log in your vote now. Thank you so much, and we will show the results in a few minutes. Okay, so we have it. Yes, yes, yes. And the last one is yes. Thank you so much. And some are still not eating biotech crops. Perhaps you will learn more, or you need to understand the safety of biotech animals. So now we will proceed to the next speaker. So can we flash in the slide for Dr. Taro Mingala so that we can introduce him properly, please? Okay, our next speaker is Dr. Claro Mingala. He's currently the director of the Biotech Program Office and the director of the Philippine Agriculture and Fisheries Biotechnology Program of the DA. Bong, as we call him, is the former center chief of the Livestock Biotech Center at the Philippine Carabao Center. He's the chair of the technical working group for the drafting of the Joint Department Circular for Genetically Modified Animals and Animal Products Rules and Regulations. He promotes biotechnology as an alternative tool to increase livestock production in the Philippines to improve food security and sustainability. He conducts biotechnology research in livestock, genetics, and breeding, health, nutrition, and product development. Friends and colleagues, let's welcome Dr. Claro Mingala.
Thank you very much, Dr. Ola, for the very generous introduction. Let me share my slides. Okay. Can you see my slides now?
Yeah, but it's not in the slideshow. Yes, it is in slide.
Okay. Okay, so the title of my presentation is all about the opportunities and regulations of the animal biotechnology in the Philippines. And it's very good that Dr. Carl a while ago already mentioned some of the products or innovations that we have that can also be adapted here in the Philippines as well. So animal or livestock biotechnology is very much into the purpose of increasing income for livestock food producers, of course, especially that animal products are the main source of protein for human consumption. And we also have doubling food production to meet the supply for demand. And climate change and disease resilience or animal resistant animals. We also have the rapid diagnosis and modern disease surveillance and proper animal management. So all of this can be achieved through the use of animal biotechnology and other innovations that can be that we can do with regards to increasing productivity and sufficiency for livestock productivity. Okay, so here are some of the specific or detailed things that we are using. For example, the marker-assisted breeding for animals across animal commodities, of course, or livestock commodities, because we are already doing this in the Philippine Carabao Center. By the way, I am also part of the Philippine Carabao Center, which we believe that this agency is the livestock research hub for the Philippines. And we are now doing some techniques with regards to biotechnology. So we are also using genome-wide associated studies, genomics, proteomics, metabolic studies. We also do some nuclear transfer activities. We use microinjections for in vitro fertilization of reproductive biotechnology techniques that we are also doing. And then all of these techniques, we use that for to enhance the productivity of the water buffalo in particular. We are also using that to assess or evaluate the milk and meat composition of livestock and the quality as well. So we are now also have some efforts on doing research for climate resilient livestock. And then we also do some studies for disease resistance. Although probably for genetic modification and gene editing, we are still on the starting stage, or we are now still doing some collaboration with other institutes and agencies that has already the experience of doing gene editing and genetic engineering. So this slide shows the new breeding innovations as the driver for change in livestock. And I think somehow Dr. Ramage already mentions about this. Using isolating the somatic cells and doing the deployment of editors for gene editing, and then the isolation of cell clones and transfer the somatic cells so that a particular animal can get the gene edited cells. No. In the Philippine Carabao Center in particular, we already adopted the somatic cell nuclear transfer technology to complement other existing reproductive tools for buffaloes. So the present work was conducted to develop or optimize a system for cloning through somatic cell nuclear transfer in water buffalo. So the buffalo clone embryos had been successfully produced in vitro, and we also trying to do it transferring the clone embryos to the surrogate recipients. But we are still optimizing this process. So this is already the part of the whole genomic editing system, which is probably we can call it on the downstream process, no? Downstream part of the whole process. So this is actually the upper stream, and if we will be able to generate a cloned, a gene edited cells that can be later on transferred to a surrogate recipient, the whole process would be realized, and we can get a gene edited livestock or animal. So it could be a different thing with regards to genetic modification and the gene editing. In as Dr. Ramage probably explained a while ago, that genetic modification is the term that usually used when we introduce a certain genetic material foreign to the specific animals, while gene editing is we only tweak part of the DNA of a particular animal that we want to improve. So this can across can we can use this across the livestock commodities. So Dr. Cal also mentioned already some of the animal biotech products that can be adapted or can be also developed here in the Philippines, such as the slick cattle, which is already developed by Axeligen, I think. It's a company in the US that produce the slick cattle. And then we have the GM salmon for the fishery sector. Comparing to the regular salmon, the GM salmon is, I think, twice or thrice the size. And then we now also have the GM tilapia, which is also developed by Aqua Bounty. And then the hornless cattle, which is being developed by USDA, particularly the team of Dr. I forgot the name. Allison, yes, Dr. Allison of the University of California, Davis. And then we have the PRS-resistant pig, already mentioned by Dr. Ramage, which is also developed by, I think, University of Missouri, and of course, the Roslin Institute in the UK. And then we have the ASF vaccine. It's a collaboration between Vietnam and the USDA. And then the GM mosquitoes and GM ticks. And of course, the muscle cattle, which is a very good source of meat. All of these are technologies that develop through animal biotechnology, some of which are developed through gene editing, and some are genetic modification. So these opportunities can be adapted by the Philippines, but of course, it's not just as simple as that. We should also be very vigilant, and we need to also put up our own policies and regulations. But for now, the Philippines has no regulatory policy or framework yet for animal biotechnology products. So as an overview, the Philippines is actually the first ASEAN country to initiate the regulatory system. It's the first ASEAN country to initiate the biotechnology regulatory system. In 1987, scientists from the University of the Philippines, Los Baños, the IRI, the Bureau of Plant Industry, the PICARD, under the DOST, formed a committee and formulated biosafety protocols and guidelines for genetic engineering and related research activities. The draft of the Philippine biosafety policy was submitted to the Office of the President, and it was signed in October 1990 as Executive Order 430, series of 1990. So the Executive Order created the National Committee on Biosafety of the Philippines, or the NCBP, under the DOST. It is composed of representatives from the different departments, such as the Department of Agriculture, the Department of Environment and Natural Resources, the Department of Health, the DOST, of course, and the members of the scientific community. So the executive order was then followed by the issuance of the Department of Agriculture AO number 8 series of 2002 as implementing guidelines for importation and release into the environment the plants and plant products derived from the use of modern biotechnology. So in 2006, the former President Arroyo issued EO 514 for the establishment of the National Biosafety Framework, prescribing guidelines for its implementation or reorganizing the NCBP. So the DAAO, or the Administrative Order number 8 of the Department of Agriculture, was then replaced by the JDC, signed by the Department of Agriculture, the DOST, the DANR, the DOH, and the DILG. On the issued circular, the DOST is tasked to regulate applications for contained use and confined tests of regulated articles. DA for those applications for field trial, commercial propagation, and transboundary movement of the regulated articles. And then the DANR for evaluation of environmental risk and impacts of regulated articles. So if you will notice, the DA, the DENR, the DOST are major players of this JDC, and of course the support of the ILG, also involved in supervising public consultation during field trials. So that this particular JDC is already revised last year, which is emphasizing that crops or plants without any novel genetic combinations are not covered in the regulatory policy. So plant breeding innovation techniques products are not covered by the revised Joint Department Circular. Now we are into the developing or drafting the GM animal regulatory policy. This particular JDC for animal products, as the subject says, rules and regulations for the research and development, handling and use, transboundary movement, release into the environment, and management of genetically modified animal and animal products derived from the use of modern biotechnology. I think the draft is already with the NCBP for final review, and hopefully we will be going. This particular draft will be signed probably later this year or early next year. Through the initiative of the Bureau of Animal Industry Biotechnology team, it's actually the time when I was there as the OIC Assistant Director in 2019. We started drafting this particular policy. The necessity to establish guidelines for GM animals and animal products was recognized. Likewise, with the JDC for plants and plant products, the proposed guidelines for animal and animal products involves the OST, DA, DANR, DOH, of course, and the ILG. So the applicability of this particular JDC, the proposed guidelines is applicable to the following. Number 1 is the genetically modified fisheries and aquatic resources. Number 2 is domesticated animals and biological products used for animal husbandry or veterinary purposes. Number 3 is biological agents used for biocontrol derived from the use of modern biotechnology and containing novel combinations of genetic materials. So it is also emphasized in this particular JDC that the products of gene editing that do not have or do not contain novel combinations of genetic materials are not covered by this circular. So for the GM animal regulatory policy, in making biosafety decisions, the concerned agencies will be guided by the principles under the National Biosafety Framework, namely standard of precaution, risk assessment, environmental and health risk assessment, socioeconomic, ethical and cultural considerations, and access to information. So the standard of precaution means the lack of scientific certainty or consensus due to insufficient relevant scientific information and knowledge shall not prevent the concerned agencies from making appropriate decisions. In such cases, the concerned departments or agencies are expected to take necessary actions to protect the public interest and welfare. So the risk assessment shall be mandatory and central in making biosafety decisions consistent with the existing guidelines such as of the NCBP and the Cartagena Protocol. So the Cartagena Protocol is actually a worldwide agreement wherein Philippines is one of the signatories. So moreover, the concerned agency shall also consider the environmental and health risk. The socioeconomic, ethical, and cultural considerations shall also be taken into account, especially on the aspect of conservation and sustainable use of biological diversity, and with regards to the value of biological diversity to indigenous and local communities. So you will be noticing that it's not only a hard science subject, but also it also involves socioeconomics and ethical and cultural considerations. The right of the public and its stakeholders to the information relevant to the biosafety decision shall also be respected by the concerned government agencies and departments. So based on the classification of regulated articles, the draft policy or regulatory policy is categorized into 4 classifications based on their intended use, namely research and development of the regulated article under contained use. I think I already mentioned this, that this will be under the DOST. Research and development of a regulated article for limited release into the environment. Number 3 is commercial use of a regulated article under containment and commercial use of regulated article for general release. Based on this classification, various agencies shall take part in evaluating and issuing biosafety permits. So for the research and development, the in-charge agency is the DOST, as I have already mentioned, specifically DOST Biosafety Committee. They will be guided by the biosafety guidelines for contained use of genetically modified organisms. For the limited release into the environment, still the DOST is in charge in coordination with other concerned regulatory agencies such as the Bureau of Animal Industry and the Bureau of Fisheries and Aquatic Resources. So since this is an animal biotech regulatory policy, the terrestrial and the fishery sector is divided into 2, wherein the terrestrial animals will be managed by the Bureau of Animal Industry, and the fishery sector will be managed by the Bureau of Fisheries and Aquatic Resources. No person or institution shall conduct R&D activities involving GM animals intended for limited release without the prior approval of the DOST Biosafety Committee. So limited release means small-scale experiments or field trials of a GM animal species performed under terms and conditions that mitigate the impacts on the surrounding environments according to CropLife International. So the GM animal regulatory policy for the commercial use, either BFAR or BAI shall lead in assessment and issuance of biosafety permits. So they are the one who will be assessing and evaluating There are 8 uses identified for the commercial use of a regulated article under containment. The feed, food, and processing for animals, xenotransplantation, bioreactor for medical and pharmaceutical uses or biofarming, bioreactor for industrial uses, animal pets shall be regulated by BAI. Meanwhile, the feed, food, and processing for aquatic species and ornamental aquatic species shall be regulated by BIFAR. For the other possible uses, the lead agency shall either the BIFAR or BI, depending on the animal species. But at present, the NCBP is trying to resolve the issue with regards to the GM mosquito regulatory process, no? Because we don't know yet which bureau will be responsible in regulating the GM mosquito. Although GM mosquito is not related to agriculture, it is more on the human health because it's actually a form of technology that can address the spread of dengue, Zika, or any other mosquito-borne diseases. So for the policy on the commercial use of regulated article for general release into the environment shall likewise be categorized into its intended use. This shall also be led by BIFAR or BAI depending on the animal species. So for the feed, food, and processing animal biocontrol or agricultural purposes and animal disease control shall be led by BAI, and of course for the aquatic or the fisheries should be the Bureau of Fisheries and Aquatic Resources. In this specific use, biocontrol for human health, household and industrial uses, the DOH shall lead in coordination with other concerned agencies. So as I mentioned earlier with regards to the GM mosquito, the Department of Environment and Natural Resources and the DOH or the Department of Health should resolve the concern with regards to the regulations of GM mosquito or any other GM insects that can be a possible vector of diseases. The proposed guidelines shall follow the general procedure. First, the applicant shall submit its requirement to BIFAR or BAI Biotechnology Office. The Biotechnology Office shall serve as frontline service and will determine the necessary actions for issuance and biosafety permits. The said office shall organize a joint assessment group. This is actually the one who will thoroughly evaluate the application. The application shall be forwarded to the JAG or the Joint Assessment Group. The said group shall be chaired by the BIFAR or BAI and is composed of personnel from various concerned agencies. The JAG will make necessary technical recommendation on the application. So the technical recommendation from the JAG shall be forwarded to BIFAR or BAI and shall be a basis for granting or denying the application for biosafety permit. There is also a statement on deregulation of regulated articles. A regulated article may apply for deregulation before expiration of the biosafety permit. BIFAR or Bureau of Animal Industry may remove the regulated article from the coverage of this circular if, based on the nature of the regulated article, the regulated article will not pose greater risk to human health or to biodiversity than its conventional counterpart. So we always consider the human health, the animal welfare, and the environmental protection. So the deregulation may be done in two ways: motu proprio, and through petition. Motu proprio is a Latin on one's own initiative. The BIFAR or BAI may consider the regulated article for its deregulation. The BIFAR or the BAI shall declare such intention with supporting documents equivalent to the contents of a petition. If through petition, the applicant, among other requirements, shall state the factual, ground why these circulars should not apply to the regulated article and published scientific literature relied upon by the petitioner. So all of these particular provisions under this proposed JDC is already reviewed, and now it's undergoing a final review. Probably a public hearing will be scheduled for this particular purpose before it will be finally signed. I think this is my last slide. Thank you very much for listening.
Thank you so much, Dr. Clara Mengala. So we have seen what are the research and development, ongoing ones in the Philippines, including in the PCC and other research institutions conducting R&D developments in livestock and aquaculture. And we have heard what's going on with regards to the regulation, and we're happy to note that it's now on the final stage of evaluation in the NCVP, and we hope it will be signed completely before the year ends. So with that, let's give a round of applause to Dr. Clara Mengala, a virtual round of applause, please, if you may. And we can now show the poll questions for Dr. Mengala's presentation. So we have 3. The Philippines is one of the first few ASEAN nations that initiated its biotechnology regulatory system. True or false? The GM animal regulatory policy is applicable to GM fish and livestock only. True or false? And products of gene editing do not contain novel genetic materials, thus are covered by the proposed joint departmental circular for GM animals. Please log in your responses now, so we'll see how much you have understood the presentation. We'll give you 30 seconds. Thank you so much. We also would like to invite you to put your questions in the Q&A box so that we can discuss them. Okay, are we getting responses? Thank you. Okay, so the first one is through. Okay, so there's still some people who are not, who doesn't know the number 2 yet, true or false. And then it's false. So it's going to, or it's true. It's going to go for fish and livestock only at the moment. And then for the last one, it would be false. Okay, so thank you so much. I'm now closing the poll. So we may now entertain questions. And first, I'd like to have our speakers on the spotlight. And let me read the first question, which was raised by the APCC Deputy Director, Dr. Caro B. Salces. Good morning. How does biotechnology techniques is evaluated in its effects to the changes of animal waste, to its effect in the soil, biological life, and environment? So this may be one of the traits that we are looking at. How will biotechnology have an impact on the changes of animal waste in the soil, biological life, and the environment? Can we also spotlight Dr. Mengala, please? Okay, who may answer that part? Can you answer that?
Yeah, I can answer that. Look, I think I mentioned in my presentation that when it comes to regulatory assessment, we look at things on a case-by-case basis. So there needs to be a scientific reason why you would want to look at the impacts of animal waste on the soil microbiota. So if you're making a change to an animal where there are conventional versions of that animal, so your comparator, there is no difference except for the trait that you're applying. Let's say that it's a thermotolerance trait, right? We know that that thermotolerance trait exists in 2/3 of the animals that are there anyway. So unless you're making a substantial change that would lead to a change in the waste product, then you might not need to actually look at what the impact might be, because it would be no different than a conventional animal anyway. Now, on the other hand, if you're making a modification where the waste that comes out the other end is substantially different, that's your target, right? Then you may want to, as part of your studies as a developer, look at what the environmental impact might be. And in many cases, if you're modifying the waste output, you're probably going to be looking at more of an environmental benefit, because that would be what you'd be targeting. So I think, it comes down to case by case. And when you're looking at case by case assessment, we look at the donor, the host of the change, and we look at what causes the change, and then we look at the phenotype, the change itself, and then we ask those questions about what is the potential impact of this change on us as consumers and the products that might come from that animal, but also the environment. So it would be dependent on what the changes were, on what you would look at from an environmental impact perspective.
Okay, yes, thank you so much, Carl. Do you have any addition, Dr. Bong?
I think there is not much direct effect with regards to the animal waste, because if ever you will do some modification or gene editing with regards to the particular animal, it doesn't probably directly affect the animal waste. But of course, if you have some modification with regards to the content, to the bacterial, microflora, wherein it will be excreted later on, that particular modified microorganism that can be excreted by a particular the animal would have a direct effect to the environment, I suppose. Thank you.
Yes.
And it could be positive. So, for example, maybe the modification means that the amount of nitrogen that comes out in the waste is less. So then you'd be asking the question, well, what does the effect of a decrease in nitrogen have on the paddock in which the animal is being grown? Now, the question then is, well, does reduced nitrogen have a detrimental effect on the environment or a positive effect on the environment?
So it's a case of basis. One of the biotech animals that is in the pipeline, I think, is the Environ pig, which would affect the animal waste. Maybe you can just discuss that as well, Carl?
I think that's the same. What we're talking about here is the same thing, is that if you're modifying through a modification to affect the waste output, then you would be looking at what that impact might be on the environment, as well as the impact on the consumption of that animal as a food product. When you're looking at what the environmental impacts are, you would be comparing the waste output from that animal compared to a comparator, which would be a non-modified version. So in that case, what you're trying to do is reduce, actually make the waste, the animal more environmentally friendly. So you're actually would be demonstrating that it actually has a beneficial environmental impact rather than a negative impact.
Wonderful. Thank you so much. There's another question. I'm just reading the questions from the chat box currently, but I'll move to the Q&A box in a little while. So this is from Dr. Clarete. This is for Claro. How is the consultation on animal GM-JDC going on? Are there any objections to it that you know of?
Yeah, there is no, we haven't done the public consultation yet, Dr. Clarete. It's now being reviewed by the NCBP with regards to the technicalities. But after that, as I mentioned earlier, we have some issues with regards to GM mosquito. So once it is resolved, we will be calling a public consultation, and we will see if there will be some resistance later on. Thank you.
Wonderful. Okay, I'm now in the Q&A box. Participants, please put your questions there. So I'm now reading the question from Rio Milem. I'm working on identifying and hopefully breeding thermotolerant livestock species. How can you help me encourage the regulatory office to accept and support this technology in the Philippines? Okay, Carl.
Well, I think Clara's outlined the process that's being undertaken to develop a framework to assess products on a case-by-case basis. So I think what you would be looking to do is, when there's a public consultation, is that you get involved in that consultation and that you engage and put forward your views. But bearing in mind that the regulatory frameworks are science-based, so you need to make sure that what you're putting forward are science-based arguments. The benefit in itself is not enough. Remember that biosafety is around the safety of those products for people and safety to the environment. So we know that thermotolerance is an animal welfare trait. That in itself is not enough for a regulator, but it's certainly enough of a pull for a consumer to want to know that the animals have a happy and healthy life, because we know that animals that are happy and healthy are actually more productive and have a more beneficial footprint on the environment. So my initial recommendation would be engage. So put forward your views and get involved in the process.
Wonderful. Thank you. You have your ideas as well, Dr. Mingala?
Yes, actually, one example of thermotolerant livestock is the one that I mentioned, the slick cattle. And I think it's already, am I right? It's already approved in the US. So with regards to regulatory system in the Philippines, we also considered some of the decisions that the other countries are made. And then we also then assessed with regards to the framework that we established here in the Philippines. So I think there would be not that much problem with regards to accepting the technology once it's already approved to other countries. So we will just be like getting some complementary assessment based on the framework that we have here.
Yeah, and I think the important thing there is when it comes to assessing those, we need to be a bit careful that we don't, because there's an approval in the US doesn't mean that it will be automatically approved in the Philippines or another country. You've still got to go through your regulatory process. But the framework that you're developing is around the decisions on whether you regulate, and if you do, what are the regulatory steps? And the case of the thermotolerant slick, the changes that are made, the way that was developed is that there was a genomic screen of animals that live in the tropical, subtropical areas. There are breeds that are known to be thermotolerant, and they identified a gene that is like a thermostat, and it helps to regulate the temperature of the animal. And when comparing that to an animal that lives in a temperate region, they found that there's only up to 6 different changes that could happen, but you only need to change one of them to effectively convert an animal that lives in the temperate region to have the thermostat of an animal that lives in a tropical, subtropical region. So it's really only a 1 base pair change. Now that can be done either using that gene editing process. So there's no new DNA that's introduced. You can use a template to guide the repair to be exactly how you want it to be. And you effectively change that animal that would normally be quite stressed, being a temperate animal living in a warmer region, to one that is quite happy and able to be more productive longer term. So that's the science. The regulatory questions are really about, is there new novel DNA that's been introduced? Is this a novel trait? What is the impact on the environment? And I think when you look at it on its case, as a case in its own, there's no new DNA, right? It's not novel because 2/3 of the animals that have that same thermostat live in the tropical, subtropical regions, and the impact on the environment would be less because the animals are happier and more productive and probably more efficient. So I think it's a tick, tick, tick from that point of view.
Yeah. Okay, there are several questions to Dr. Mingala on the regulations in the Philippines. I'm just going to read some of them. How will GE animals, livestock be regulated? What will be the process? It has been described a while ago. I think you have to reiterate this again. And will release permits have an expiration? That's one. And then the NCBP regulation, biosafety regulation, is highly restrictive. thus limiting several biotech R&D initiatives. Are there plans of revisiting this so that it will be more supportive of rather than restrictive? This has been addressed as well for crops and now for animals. So I think Dr. Mangala can shed light on all of these questions.
Yeah, actually, the question regarding the restrictive nature of the framework or the policy is already addressed before. That's why I mentioned earlier that the JDC for plants is already amended last year to make it more friendly, I suppose. And this JDC for animals is already patterned on the amended JDC. So I think once this particular draft will run through the public consultation, we hope that everyone would participate so that we can also hear the sides of this developer in particular. And our team is actually at the middle, and we want to really complement the developer as well as the sectors, and of course the regulatory team that will be putting into with regards to animal biotechnology, and hopefully we can harmoniously exist with regards to this particular endeavor. Thank you.
Yeah. Okay. Questions for you, Dr. Mingala. More questions. This is from an anonymous. Recently, the U.S. president released an EO on advancing biotech innovation. In it, which include international cooperation and others. It is also expected that more opportunities and technologies will arise for the biotech field due to this EO. Is the Philippines ready for this? Are our policies being reactive or pro-reactive regarding the possible technologies being developed?
Yeah, as I mentioned already, the Philippines is very much proactive with regards to this. That's why we are now drafting this particular framework. We don't want to wait until a particular activity will be implemented here in the Philippines, but as well we are putting this particular framework. Of course, the possibility of influx of biotech products that will be coming into the Philippines. So we are ready, and we hope that this particular framework will be signed soon, so that we already have the document and the guidelines that we can use for this particular activities. Biotechnology, either the process that we'll be doing here in the Philippines and the products that will be coming into the Philippines. Thank you.
Yes, we are ready. Thank you. Okay, so we have now a question on maybe this is about communication. So aside from regulation, consumers or users, such as farmers, acceptance is also a key. So what efforts are being done to improve understanding and acceptance, and what can private companies do to improve this as well? So we may have Carl also to respond to that. Let's have Dr. Mingala first, and then Carl.
Sorry, I'm not able to catch it. What was?
It's on efforts to raise understanding and acceptance of animal biotechnology. What can private companies do to improve this?
Okay.
Yeah.
Okay. Probably I will be talking more as a director of the DA Biotechnology Office. When we wrote the framework, I was the assistant director of the Bureau of Animal Industry, and it's the initiative of the Bureau of Animal Industry to do that as a regulatory agency. And now, as a director of the DA Biotechnology Program Office, we are actually helping the developers or the companies that are developing biotechnology products to somehow reach out not only to the consumers, but as well as to our legislators and the judiciary as well through advocacy campaign, and of course getting the support of other stakeholders such as the ISA and the other partners that could help us give informations to the public. So that's how we do in the Department of Agriculture Biotechnology Program Office, and we hope that we can reach more people so that they can understand more all about animal biotechnology.
How about how could the private companies, what can they do? What do you suggest they can do to improve acceptance as well? So you were talking about the public sector. I mean, you as a public person.
Yes, actually we are like bridging the consumers and the developers, and these developers are the private companies that we have not only in the Philippines but also the international companies that are planning to export some products into the Philippines, or even the technologies that they have. So we are actually kind of bridging them, and we are also facilitating so that the informations that they have, probably the documents that they need to submit to the regulatory agencies would be more what you call this, understandable, and facilitate as well the regulatory side for them.
Yeah. Okay. Thank you. And you have any thoughts also, Carl?
Yeah, I'll take a slightly different approach. And I think the first thing that you want to be thinking about if you're a private sector organization wanting to introduce a new biotech product is the first thing you have to do is acknowledge that some people are going to be concerned. Some people are going to not be comfortable with what you're putting forward. So in some ways, part of it is about, one, acknowledging that concern, and then being open to discuss those concerns with the people that may have some questions. So what we're starting to see is a shift in that the traits and the products that are starting to come through are not necessarily targeted for farmers. They're not necessarily productivity traits. They are traits that are around things that as consumers we feel good about. The welfare of animals, for example. We want to know that an animal had a happy life to produce the products that we eat. We want to look at alternatives, such as there's a bit of a swell in alternative to animal proteins from animals, but animal proteins from fermentation or from plant sources. So the first thing is to acknowledge people have some concern, and as a company, your number one goal is to build and establish trust with your consumers. And the rest of your supply chain. So when I'm working with my clients, we look at the continuum from developing your freedom to operate, your regulatory approvals, your proof of concept, your intellectual property, the social license to operate at the other end, which is getting the consumers on board, engaging, talking about your product and showing that you care. And in the middle is what we call the permission to operate, which is your engagement with the supply chain, with the other industry stakeholders that will be involved in delivering your product to market, because not all developers are vertically integrated. So you need to get support all the way along your value chain. And that starts with engagement and conversation. And webinars like that ISA run, for example, is a great way of engaging with people and giving information, providing opportunities to talk about the technologies and feel comfortable being able to answer questions that you might have and raising the concerns that you might have. And then lastly, as a private sector organisation, when there's an opportunity to be involved in regulatory reform, then you must get involved in that regulatory process and be happy to engage and to offer, your support to the regulators in shaping and developing a framework that not only satisfies the role of government, but also supports private enterprise.
Wonderful. Both of you have given very good examples of how to do things, and it should be stepwise, and trust and transparency should be there as we go along so that acceptance will be, will be there. Now, I'd like just to announce that we have already posted in the chat box the post-webinar form, and if you can respond to the survey, you will be provided the certificates and also The Zoom recording of this webinar and the PowerPoint presentations will be at the website, at the ISA website. We're still entertaining questions, so please post your questions. And now may I read the question from Dr. Clarete again. So this is for Dr. Claro. Will the regulatory structure for animal biotechnology follow the pattern in plant biotechnology regulation, wherein the NCBP had declared that only GMOs will be covered by the JDC on animal GMOs, and the DA will come out with a circular on animal breeding innovations not using foreign gene? Is this correct? Yes, please go ahead.
Yes, it's correct, and it's I think almost similar to the amended JDC for plants.
Okay, yeah, so that's clear enough. And we have seen that as Dr. Clara mentioned or explained a while ago, it really followed the one on plants. Do we have any more questions? Okay, there are a few more questions in my head, if you may. What do you see the Philippines? This is for Carl. You have been listening to not only for crops but also for animals. Really looking forward for commercialization, the innovation products, and gene editing as well as animal. And even if we say that we are in the one of the top countries in Asia, what are the other things that you think is missing? We're still slow, but the regulation is there. The scientists are really supporting the technology, but there's still something missing. What could that be from an outsider looking in?
Well, thanks for that really difficult question, Ola, because I think what you've highlighted are some very important things that shouldn't be forgotten, and that is that the leadership that the Philippines does play in the region in terms of its regulatory frameworks around biotech, and has done a sensational job in providing clarity for developers around new breeding technologies in crops, and is now doing that for animals. And Clara is doing a sensational job at facilitating that process, which is not easy. I guess the problems that you have are the same everywhere, and that is that a lot of the technologies that really have promise are being developed within public sector organizations, and the challenge is transferring the technology from public sector to private sector to commercialization. And the commercialization of biotech products is incredibly challenging. And it's challenging on a number of fronts. One is that often the investment is there's quite a lot of investment required to get a product to market. But a lot of that is because the technologies seem to be being pushed to market rather than pulled to market. And so I think, like everywhere else, there's some really good science and some great products, but they're not necessarily commercializable. What you need to be able to identify is, what are the products that people need? What are the products that people want that are going to pull for something through? Recognizing that biotechnologies are just tools. Mm-hmm. There is some superb breeding technologies being used at the Caribou Centre just to do normal selection, understanding productivity and production systems for livestock. All of those things are incredibly important as well. Biotechnology is just a tool. So it's really about identifying what are the products that are really required to solve particular problems. Now, we know, for example, that climate variability is something that many countries are now really starting to address, and agriculture is always seen to be as a problem child when it comes to emissions and its impact on the environment. So that's the opportunity. So what are your public sector organizations doing to put forward solutions to that perception? So that could be in cropping, in creating more digestible, high energy inputs. It could be developing animals that are happier and have a less environmental impact by being able to survive in warmer temperatures and produce more. Those are the sorts of things that really need to be addressed, and facilitating and helping public sector technologies to market. That's where I think the big challenge is for the Philippines, but it's also the same challenge everywhere else.
Okay, yeah, that's correct. Do you have additions, Bong, in response to those suggestions by Carl? I mean, what are we doing with regards to those suggestions?
Yeah, actually, doing activities on animal biotechnology is very challenging compared to crops. So probably experts-wise we have, but resources are very limited with compared to crops. We have a very limited resources here in the Philippines, and it's not that easy. There's a nature. There's a kind of very big difference with regards to the nature of crops and animals. For example, grafting, you can just do the grafting in the crops, but in animals you can't do that. You will be sued by the animal welfare activists. And it's really very challenging. So probably the Philippines would be like wait and adjust to what other countries have been doing, and then probably can start from there, getting some technologies and try to do here in the Philippines. I think that's my perception.
Okay, thank you both. So this is a question again from Dr. Claretta. This is for both of you. In terms of time and R&D resources to develop the biotechnology, is animal biotechnology more difficult to develop than plant biotechnology? I think
we already, yeah.
Dr. Claretta, I think this is okay. It has been responded to by both our panelists. Now there's a question on health here in the Q&A. More and more vector vaccines are now produced through bioengineering, such as AstraZeneca vaccine from British company using adenovirus. Do animal vector vaccines need to be assessed by BFAR before they are marketed? This is for Claro.
Animal vaccines.
Yeah, the animal vector vaccines.
Animal vector vaccines for human? If it's for human, I think it should be the FDA. Not the agriculture,
not the BFAR, but if it's for animals, it's for
it depends on, like, for example, as I mentioned, if it's terrestrial, it should be the Bureau of Animal Industry, and if it's for fisheries, it should be the BFAR.
Yes, and then there's one question here: is GMO product safe? It has been presented by Dr. Carl Ramage a while ago, the safety of biotech crops, not only the crops, but also animals. So I hope that's clear already.
Yeah, I think the point there, though, Ola, is that the default position that is generally taken is that we don't know. So our regulatory system is designed to be able to address the question of whether they're safe for people and safe for the environment. So I don't think we can just answer the question, is it safe? Because there's a lot of questions around that. I think what we can address is that we have robust regulatory systems that allow us to answer that question. And the Philippines has had a very robust and very successful regulatory system for a very many number of years, which should give the public confidence that if the Philippines regulator says it's safe or that we don't need to regulate it, then we should trust them, because they have a long history of demonstrating safety assessments in products derived from biotech.
Okay, those are very good answers. I don't see any more questions. So at this point, maybe we'd like to have the last few remarks from okay, there's one more question. Okay, this is the last. In Europe, there is a strong resistance against GEGM. What are you seeing, and are there any signs of them considering otherwise, especially if technology is used to improve human or animal welfare? Okay, this is for both of you. What are your thoughts?
Do you want to go first, Claro?
Yeah, for me, I don't know. Probably that's their choice, because if they really have a very good or a science-based regulatory system, they should see the pros and cons and not just put in strictly no for GM or GE products. So I think it's the EU's choice. I don't know really where they're coming from. Thank you.
Yeah. Look, you know, it's always a challenge, isn't it, when we look at another area that is perhaps a trading partner where they have quite different views to our own. And you know, starting to see a shift in particularly the UK, starting to move because they're no longer part of the EU. They're starting to look at their sustainability and their ability to be competitive and to feed their people. We have seen from in many countries coming out of the pandemic a shift in policy around more local production and not relying quite so much on imports but also the effects on exports. So whilst we sort of see the EU as a challenging jurisdiction for regulations in biotech, we're always ever hopeful that things will change, right? And at the end of the day, it's their choice. And if they are a market, then we need to satisfy the market. If they're not, then it doesn't matter, or we find another market. And I think we're starting to see that, particularly out of Africa. The African nations are starting to move more towards accepting biotechnology and not necessarily listening to the EU, and starting to look at, because of things like the impacts of African swine fever and fall armyworm, these are dramatic impacts on the economy of certain countries. And so therefore that drives political change to say, well, what tools do we have? What are the solutions to these big problems? The EU, there's many, many countries, but each of them have their own problems. And so at some point, the only solution might be a biotech problem solution. So we'll just have to wait and see. I'm never really worried about the EU. They're too far away.
Okay, I'm just going to read a comment from our mentor, Dr. Violeta Villegas. I think the essence of safety assessment is to show that the GM or gene-edited products are as safe as the conventional counterpart, which is true. Yeah.
Correct. Yeah, and the foundations of our safety assessment system through Codex is about exactly that. So we look at the donor and the host, and we compare that to a comparator and demonstrate that except for the trait of change that we're intending to change, is substantially equivalent. And I think we have a long history of being able to do that and showing that we can do it safely and effectively. So yeah, I fully agree.
So I think I'm going to close the discussion now, and may we have your last few words. We start with Dr. Carl Ramage, please.
Look, I think we covered off some really good questions there, and it's been great to see the engagement through this webinar. I guess I'd just like to reiterate two things. One, that there's an opportunity coming to be involved in shaping the framework through public consultation that Clara mentioned. And so if you are interested, I think people should engage and be part of the process, because there's no point standing back and then complaining about it afterwards. You have an opportunity. The other one would be a bit of a curveball, which is, and I'm not sure if it's in there, Clara, but timeframes. So for public sector and private sector organisations wanting to take a product to market, having a time frame for a regulator to make a decision is incredibly valuable, because it creates certainty in terms of understanding the regulatory process. So we're very lucky in Australia that both our food feed and our cultivation regulators have regulated timeframes to undertake their process, and that is incredibly valuable. So if you don't have timeframes built in, build them in.
Okay, wonderful. Yes, I think that we have. Please.
Yeah, okay. For me, the Department of Agriculture is very much into using animal biotechnology, or not only animal biotechnology, but biotechnology in general, because this is an opportunity or a tool that we can have to increase the productivity of our agricultural commodities, not only crops, but also livestock. So we hope that we can harness this particular technology so that we can use this, of course, for the benefit of our people in the Philippines. We all know that there's a lot of things going on economically wise. There's a high increase in prices and low productivity. So we should take this opportunity to take the biotechnology as a tool for agriculture. Thank you.
Thank you so much, Dr. Clara Mingala and Dr. Carl Ramit. Thank you so much. Now let's move to closing remarks. And before I do that, I'd like to invite our participants to please respond to the webinar survey so that you can have we can see what you think of the webinar, and we can learn from your responses. So thank you so much, panelists. Dr. Caro Salces is the Deputy Executive for Administration and Finance, Executive and Finance of the Philippine Carabao Center, who will give us the closing messages. The floor, Dr. Salces, please.
Thank you, Doc Rodora. Am I clear?
Yes, sir, you are. Thank you.
Okay, thank you for this opportunity to be with the group. Thanks to the USDA for this Base Safe program, and also to your group, Dr. Almidita, for hosting this program. I'm also educated. I'm one of the 80 plus participants who are educated with this tech program. So my thanks to our speakers today, Dr. Carl and Dr. Claro Mingala. Ako naman ay si Dr. Caro. So naging homonyms kami na. So for the basic program of USDA for promoting these opportunities in biotechnology in answering the food demand of the world. So this is a very challenging problem that we are facing in the next 50 years. But again, the biotechnology could be an answer to the question of food demand of the world. So to Dr. Carl for showing those success stories in biotechnology in the world, and also assuring us that it is safe. So Dr. Mingala, our own Deputy Executive Director, and also the Director of Biotechnology in the Philippines, for sharing to us the regulations that the Philippine government is now putting up. So that assures us that the biotechnology system that we will be adapting or will be allowing to come to the Philippines, that we are safe. The Philippine Carabao Center is one of the pioneering agency that adopt biotechnology in its development efforts now. So we were able to develop the Philippine dairy buffalo through biotechnology systems, and also in product development and animal health. So thanks to our group headed by Dr. Mingala. Again, the opportunities of biotechnology in answering the food problem in the world is very important, but it is not without challenges, because you have seen some objections in other parts of the world. Sabi niyo, it's in Europe, and also some parts, some areas in the Philippines where there's a challenge in adapting biotechnology. But I think it's sabi natin, it's science-based, and we answer those questions in terms of science also. So the challenge is also how to prove that it is safe. No, in my first question kanina is how do we measure, no, in terms of environment, the biotechnology processes or biotechnology products that we adapt in the Philippines, in terms of the environment. So I think that was clearly answered, but in some areas, because when we change some part of the food chain is also affected. No, so I think if there are questions in biotechnology techniques that we implement, we should answer them in terms of biotechnology research. I have not seen more research on. We have more research in animals, but maybe important biotechnology research in the soil component, no, is also important to complete the value chain, that we will be assured that the food chain will be maintained or will be improved. No, so again, I'm one of supposed to answer on kumakain ba ako ng biotechnology food products. Yes, of course. No, we are, hindi natin alam that we consume many biotechnology products, because corn alone is in the Philippines is a product of biotechnology, and we are eating a lot of corn products now. So I'm one of the consumer of biotechnology products, and I believe that it is the answer to the food problems in the world. But again, it is also an opportunity now to develop further our biotechnology systems in the Philippines, and the Philippine Carabao Center as an agency is one of the pioneering agencies who adopt this technology. And I guess with the help of our experts, we can further promote biotechnology in the Philippines and its actual practice in the farmers field, as it answered problems in food problems in the Philippines. Again, thank you sa ating mga organizers, to our speakers, and also to the listeners nationwide. This webinar has provided us much information, and there's a need that more webinar series like this can be done to educate the people, and also to promote related studies in this aspect. I think this is the way to go. Now, again, thank you. Good morning to everyone.
Thank you so much, Dr. Salces. So with that, we would like to thank our panelists again, and also for the support of Be Safe. And we hope that we can engage with you again. We will be holding the next webinar in around October or November, which will be on Bidaville. So we hope that you will be there again to support us and to learn some more things about biotechnology in the Philippines. Thank you so much. And I hope that you will respond to our webinar survey. Thank you, Carl. Thank you, Claro. We'll see you again. We'll have more of this engagement. And thank you, Dr. Salces. Goodbye. Mwah. Yes, thanks.